But I'll be talking today about work that I started as a graduate student back in Arizona and have continued since starting at Penn State Harrisburg. Okay, great. Okay, so probably don't need to tell folks in this room, but there's a striking degree to which microbial communities are shaped by which can affect everything from interactions within that species, interactions with partner species in the case of symbiosis or hosts, and cooperation can sort of, in the short term, strictly an ecological sense, by influencing the structure and function of these microbial communities across a diversity of environments, and also in the longer term is an important outcome, but also driver of including adaptation and coevolution with partners and hosts. And again, there are certainly folks in the crowd who probably have a lot to say about that. Now, the idea of cooperation microbes itself isn't necessarily that new, or the observation isn't necessarily that new. For 50 years now, there have been instances of really striking levels of cooperation in microbial lineages like the also in some other remarkably similar appearance that are phylogenetically completely unrelated, like the ciliate bacteria. So we've known for a while that at least certain microbial lineages are capable of striking cooperation, including here self-sacrifice in the case of. But what's more recently is just an appreciation of the extent and the importance of cooperation in microbial communities. So this is a cartoon of the very simplistic view that is dominated for a long time. It would be a bacterium, as these things that are off on their own. So every cell for itself, swimming around, relatively simple, and learn more, including things that we would consider relatively familiar, things like E. coli. And we find out that if we allow these things to have any sort of self-determination, if we prevent them doing any, growing them in mixed cultures, for example, but as soon as you arguably most, I'm not sure exactly how to answer that, but we'll do something a little more complicated. Now this is a schematic based on certain lineages. It's unclear the degree to which the multicellular structures are created by these microbes for conditions. It's unclear if there are any sort of central tendencies. In some cases, these are just simple multicellular aggregates, flocks, things like that. But certainly in some cases, some well-studied cases, there is a set of behaviors that underlie the emergence of a multicellular structure. I'm dancing around the word development because that's, of course, a big word if we're going to use it in this case. There are people who argue that we should be viewing this at least as a form of proto-development. Which really speaks to the complexity of the interactions among these microbes. Again, because development is by its nature a multicellular process. It involves coordination, communication among different cells. Oh, and I should just also say, if anyone finds anything problematic, wants to clarify anything, please just raise your hand or jump in. Yeah, so. Please do that because sometimes I start moving. Okay, one great example and well-studied example of is this general class of traits called allelopathy. And this term allelopathy simply means killing others. Essentially, it's a mechanism from the plant world. where a plant will release some kind of a chemical compound that will inhibit competitors, basically other plants from germinating near it, and therefore, it monopolizes the. It gets a little more problematic in the case of microbes because, again, in an unstructured environment, especially, doesn't have the same sort of stability, environmental stability, as something like a larger plant. If a plant releases in its neighborhood, then it will continue to benefit that plant by inhibiting germination, again, very close to that cell, or that plant. So that is, there's a very clear connection level fitness or individual level performance in that case. It gets more complicated with microbes simply because they are plants. At very high densities, they tend to overgrow each other, and in at least the initial conception of microbes, we would often think of them as being in relatively more dynamic environments. And so then this raises questions, specifically about the evolutionary stability of something like allelopath. So if a chemical is secreted by a cell, and or some amount of these chemicals are secreted by a cell, and the benefit of those is reduced competition, then what's to stop a neighboring cell that's also resistant from benefiting from that trait without investing in the same way? And so this problem is sometimes referred to as cheating. So it's not restricted, of course, to allelopathy. It's something that anywhere you see cooperation in, well, I was going to say in society, there is at least a potential for something like cheating. And so this is a real question. And one way that people have gotten at this historically is to borrow from economics, which isn't uncommon, and specifically borrow from this branch of economics called game theory. And so this is, I believe, this probably the simplest and most widely known, at least I think, example of a game from game theory. It's referred to as the public goods game, but in the two-player version, it's also called the prisoner's dilemma. So probably you've heard maybe the latter one might be a little more widely known, but they're essentially the same thing. And so to move through fairly quickly, the benefit of taking this approach is that you can distill potentially very complicated things, processes, down into a very important and very, very simple core that you can then use to understand, if we can have insights about how the public goods game works in general, then we can look for ways to map that onto real life situations to try to understand, first of all, the problem of cooperation, which is what the public goods game illustrates, but then also potential solutions to it. So to walk through how I'm going to represent this, there are two players, player one, player two, and in the middle of this little box is just representing the public good. So you can think of any of your favorite examples. In the case of humans, it could be something like infrastructure, and we invest in the public good by paying taxes. In the case of microbes, it may be an investment in a costly competitive compound that's being secreted outside of the cell, potentially benefiting others. And so the benefit itself would actually be not just toxins, but would be the reduced competition that results from those toxins being produced. So to step through kind of slowly in the first one, I kind of skipped over this. The two strategies in the public goods game are simply, when it's just a single round, are a player can either cooperate or cheat. And what that looks like is cooperation is an investment in the public good. So I've chosen these numbers from nowhere, except just to be convenient and easy. So in this case, to invest in the public good requires an investment of 4 units, whatever that means. So player one invests, and in the best case scenario for everybody, player two also invests. The total investment comes to 8 units. And then, an important thing then is then that investment has to appreciate. That just means the benefit has to be worth the costs. So you multiply, the total investment times some number that's greater than 1. And in this case, again, I've chosen this just to be convenient. And at the end of the round, the total payoff, through 12, is distributed equally to all players. So two-player example, you can do this with all the other, with you know, any number of players, essentially. And so the net payoff is 2. They are both ahead 2 units, whatever that means. Things get more interesting if you run through this thing, but player 1 cheats. Because player 1 avoids the initial investment, but still benefits equally to player 2 at the end of the round. Meanwhile, player 2 has done the same thing in both of these situations, but only gets half of the payoff because player 1 did. At the end of this round, if you can do the math, then the cheater is way ahead of the cooperator. And not only that, the cooperator is behind, of course, how the cooperator would have done had their partner cooperated, but also this is a negative. You know, they've lost. They should have just stayed out of it. And the most simple to understand probably is if both players decide to defect, then no one gets any payoff. Everything's neutral. Okay? So again, very, very simple, but it captures some kind of important dynamics here. So you know, the first thing, what we might expect is cooperation is good for groups. More cooperators means a higher mean payoff. But this is the part that is really challenging. I was just teaching this in one of my classes, and it's remarkable how hard it is to get humans to think past the intuition that it's good to cooperate if your partner cooperates. That's you know, so I try to frame it as cutthroat as I can, and still it trips people up because it just makes sense to us. That itself is interesting, but human evolution is a different topic we're not going to be talking about today. In any case, in terms of the actual payoffs, if you compare, if you look at these different groups, if you compare the performance of an individual who has a cooperating partner, you see that the defector comes out ahead of the cooperator. If you do the same thing, if your partner then defects, and you compare these two, then it's still better to be a defector. It's always better to be a defector. And that is the public goods problem. That's not a solution. It's just encapsulating the challenge for any sort of evolved cooperation. How can it overcome the immediate selective advantage that we would expect to be favoring cheaters? Okay? Any questions so far? Hopefully pretty, yeah. And so this is the implication, that competition can actually destroy cooperation in the public good, even when public good would benefit ultimately all the individuals. So how do we explain, you know, we know that antibiotics came from somewhere. So how do we explain the evolution of cooperation in, for example, through allelopathy in microbes? Well, it turns out a bunch of people have looked into this, and if we insist on keeping our bacteria in liquid cultures, then there is no solution to this public goods problem, and cooperation does collapse. It's invaded by resistant, non-toxic mutants that are able to basically take advantage, even in an environment where there is competition, where allelopathy is providing a population-level benefit. Even then, you see invasion of non-toxic resistant cheaters. However, that outcome can be reversed if you grow these same bacteria in a structured environment. So Petri dish isn't necessarily a native habitat for bacteria, but something like a biofilm, which might be a little more natural, can also serve the same, well, can actually have the same effect as the highly artificial structured environment that you see here, by specifically keeping closely related cells together, but also reducing the loss or the diffusion or mixing of the secreted allelic chemicals, keeping them localized. Okay, so I certainly don't mean to imply that this is a done deal and that anyone who's working on this can go home early. There's a lot to be done still when we're thinking about bacterial allelopathy. Lots of fascinating work coming out even right now, especially about the different mechanisms that can underlie competition, interference competition within microbial communities. However, in general, from a bird's eye view, it seems fair to at least link the potential for allelopathy in biofilm-forming bacteria to the biofilm lifestyle. So self-structured environment And that's part of what's interesting, is that itself can evolve. But in general, that makes sense. Toxic algae blooms are a different matter because they are not at all like biofilms. And on the face of it, they would seem to be acting in a very similar sort of way. That is, they are collectively, a toxic lineage is collectively suppressing its competitors. Not only competitors, also predators. And having enormous ecological success as a result, as evidenced by, this is a red tide, and this is the golden tide I'll be talking about today. But you can see these from a long way away, and of course, I think even still right now, this Carenia brevis is a major problem right now in Florida. So, the fact that this is not only evolutionarily stable, but quite ecologically successful, it's hard to dispute. And although we lump these things together, just like we use the term biofilms to refer to a lot of different things, a toxic algae bloom can be produced by many, many, many different things that are phylogenetically very, very divergent. So around here, I would say we're probably more used to cyanobacterial toxic blooms. These are things you find more commonly in freshwater environments, just a few different Anabina and Microcystis, a few different prokaryotes that are involved in toxic blooms. But this term also encompasses very different eukaryotic groups as well. So these are haptophytes in the diatoms and diaflagellates. So a great deal of diversity, which, you know, evolutionarily I suppose is a bit interesting, but hopefully also underscores the fact that this is not an odd thing that we've stumbled upon, one particular lineage, one particular environment. It affects environments across the planet, from ice-covered lakes to farm ponds to Lake Erie in Florida. So to begin working on this, I started working on this in graduate school after I made some sort of a statement to this effect in, I think, my second year. In a departmental seminar, I made a very strong statement about spatial structure is absolutely required. You'll never get allelopathy without spatial structure, something like that. Just being that guy. And my eventual advisor, he wasn't at the time, Jeremiah Hackett, challenged me about this. He said, well, how do you explain this? And I wasn't able to, and it was frustrating. So we began working on this species, Prymnesium parvum, it's often called golden algae. And beyond being a persistent problem in many coastal environments, especially it's associated with aquaculture, it's also become established in the US back in the 80s. This is when the first bloom was reported in inland waters. It's primarily a problem down in the Southwest. However, it does bloom throughout the country. I think a majority of states have at one time or another experienced a bloom. And these blooms are very destructive. That includes Pennsylvania, by the way. Dunkard Creek, bloom 2014, I think. It was very, very destructive. And these are very destructive. There's just another picture of parvum. These are very destructive because they kill fish. And they don't just kill fish, they kill all the fish, when densities permit. In this case, these are dead fish along here. These aren't rocks or anything. I keep coming back to this figure. Last year, someone from the Texas Parks and Wildlife told me that they removed 17 dump trucks full of dead fish from one lake, which is, first of all, I think that should be like a metric unit or something, but it's remarkable. And it happens every year. There are bodies of water they've just stopped trying to restock because every year they just stop again. And it's not just fish. It's pretty much it's a vast majority, at least, of animal life in these hard hit communities. Okay, so consistent with the idea of allelopathy, yes, collective toxicity is an important factor in blooms. Triggers may be a slightly strong word, but it certainly is involved in any of the noticeable effects that blooms have in the toxic species we're talking about. Of course, algae does not have to be toxic to have destructive effects because of anoxia, dead zones, those sorts of things. But in the many cases that I'm referring to today, at least, this is a common denominator despite all the phylogenetic diversity. And basically, the bloom and toxicity are the result of a positive feedback between the density of the potentially toxic cells and their ecological strength, their ability to reorganize communities and eliminate natural predators, which can include competing algae, harmless algae, in addition to various kinds of grazers or predators. And so we return to this issue then, how do they do it? Given that there is, would seem to be quite a potential for the problem of cooperation to interfere with this little scheme they have going on. If the toxins are costly, these are things we should be able to empirically test, of course, but if the toxins are costly, and if the benefit is evenly distributed among all cells, then we would expect to see cheaters. And yet, at least for parvum, much to my frustration back in my second year, I couldn't find any. There was one instance in which reduced toxicity was reported, but they didn't keep that strain. So there hasn't been a great deal of interest in that or effort in looking into it. So given that, the first thing to do was to actually look for them. And so I got some samples from Texas Parks and Wildlife, just started sampling or isolating different lineages that anything I could get out of these samples. And then measure toxicity just through a hemolysis assay. And the things that I was looking for in this order, how do you actually define a cheater? How do you know when you see one? Well, the first thing would be, do you see an absence or a dramatic reduction in toxicity? And does that actually translate into a benefit? Is there a benefit from avoiding the cost? I guess, yeah. So in other words, is toxicity costly? But then that alone is not enough. That alone would be consistent with maybe just adaptation to a new environment or something. But if this putative cheater, if we were to find one, if it's also able to benefit from neighbors that cooperate, that's a really crucial thing in developing this thinking. And finally, it has to also outcompete the cooperators. It has to have a selective advantage over cooperators. So to begin with, first of all, do we see any kind of variation in these populations at all? So these are these data on the y-axis. This is toxicity. It's just a measure of toxicity based on a bioassay. It's basically log ratio of a prey alga that's killed per potentially toxic alga. And so the things that are shaded in gray, that's just the things that henceforth I'll be referring to as toxic based on this. The non-toxic strains or isolates putatively non-toxic are shown here in white. And the thing, so again, these isolates we characterized quite a while ago. It was much more recently, actually just this year, with Jess Bauer and others in my lab, that we were able to finally get around to testing the new isolates, being from the year after that. I just hadn't had a chance to look at yet. And so I was pretty excited to see, not only do you see certainly a very low toxicity strain in mixture with a high toxicity strain, these two from the same sample, but we saw it again in the following year as well. So in general, it would also be good to know, potentially thinking towards mechanisms behind this. Perhaps these could be explained in different ways, but blooms do have a substantial genetic diversity. So this is still our kind of early stabs at trying to quantify diversity, and it's really more of a yes or no. This is based on the ITS1 sequences of the different isolates, and that's how folks have characterized these other isolates over the years. So at a coarse resolution, there does appear to be substantial genetic variation in these blooms. We've left a lot of them out of here just for readability. But I should say that also my collaborators, I really need a picture, Jen Weiskaver, who's at Purdue, we were both in the Hackett lab at the same time. So she has her own lab now at Purdue, and they're doing some really exciting work with these strains and others, where there's finally sequencing the genomes, not finally because I'm just amazed there aren't already genomes for these. So they're working on genome assemblies, and they are also going to be doing some transcriptomics. And I don't want to say too much, but you know, the preliminary data sounds really exciting. Like there's, yeah, from a genomic perspective, it's not like there's one or two SNPs. It seems like there's a lot more happening. But I need to find out exactly what I can and can't say, because that just sounds too vague. But anyway, it's hopefully they'll be publishing that stuff before too long, and then I can say much more about it. Anyways, so certainly a lot of genetic variation in blooms, in addition to heritable, stable variation in toxicity. So does that translate into an actual phenotypic or growth advantage? And this was work that wouldn't have probably been possible without Noelle Espinosa, who was an undergraduate at the time. She's since gone on, and she's I think nearing the end of her PhD at the University of Arizona. But, so yeah, the question then, is there an actual advantage to underproducing toxins? Well, so in this graph on the y-axis, we've got cell density. It's not log transforming, just cell density. So when growing both of these things alone, just under normal conditions, phosphorus limited, everything here will be under phosphorus limited conditions, unless otherwise noted. So if you just compare these things, a highly toxic strain grows to a much, much lower density than the low toxicity strain. And that's in general what we've seen for other isolates. I've just focused on one here. So yes, the final densities of the low toxicity strain much, much larger than the high toxicity strain. And when you mix them together, this is the density you have. In general, we still don't have a very, very good method to distinguish the high and low toxicity strains in mixture. And that's a problem. It's hard to overemphasize how little there is in terms of tools for studying these things. You know, we can't transform them. Yeah, Kevin.
And the toxin that's produced is not toxic towards itself. Like the minus toxin isn't affected by it.
That's a good question. It appears to be no. Yeah. But that's another thing we know frustratingly little about. But maybe we'll get to it more in a minute. Okay, so as a preliminary sort of way of seeing how things may be shaking out, this particular lineage was substantially larger than this lineage. And that's actually why I chose these two, because they could be fairly well differentiated on the basis of size. There are also high toxicity smaller cells that are around the same size. And actually, since the more recent isolates are reversed, we have a very highly toxic, very small cell, and the reverse. So at first I thought maybe size was driving it, but it doesn't appear. In any case, I'm just going to verbally describe, these appear to be, these populations shift towards being smaller in general. Very coarse way of characterizing it. But they do shift towards being smaller over the course of the experiment than at the initial point. Okay, so it appears that there could be at least an advantage to avoiding the cost of toxicity. And that was in an environment where there was no reason to produce toxicity, right? There's no benefit either, so to distinguish the two. So in order to try to understand if there was a benefit from toxicity that could be exploited by the non-toxic cheaters, potential cheaters, then we looked at the competition of this species with a diatom that we isolated from nearby waters. And diatoms are very good competitors in general. Broad statement, but diatoms are pretty solid. They're pretty common in most freshwater environments. So this is how this diatom did by itself. Grew okay, but then when you mixed it with a monomorphic non-toxic strain, its numbers were certainly reduced, but nowhere near as much as they were if you had a toxic strain that was present. So monomorphic or mixed populations both saw dramatic reductions in the diatom densities. So how did this affect the parvum populations? Well, here again is the non-toxic strain by itself. You'll see it takes a big hit from competition with the diatoms, maybe not shocking. The toxic strain, in contrast, is not harmed at all by diatoms. That's not a significant increase or anything, but certainly they are being harmed by competition with diatoms. So far, this is just kind of consistent with our expectation from the public goods problem. When you mix these together, Also, squinting at this and squinting at that, and then comparing this, it might not be shocking, but we also saw a decrease in size of the parvum population over that time too. So what? Yeah, question.
Is the toxin production at all responsive to the presence of the competitor? Does it ramp up toxin production in that case?
That's a good question. I will tell you that as far as we've seen it, the primary environmental trigger for toxicity seems to be nutrient stress. But maybe I can try to return to your question towards the end. As we're finding out more about it, there's reasons to think that could be kind of an interesting question, besides even just this. Yeah. Any other? Okay, so now what? We are stuck with cheaters. We don't have any good explanation for really how these blooms are occurring, really, from a broad perspective. So we have really two major problems. One is the evolutionary problem we've already described, problem of cheating. How can there ever be selection for toxicity in this context? But there's also another very serious ecological problem. And that's because allelopathy is positively density dependent. There's an invasion from rarity issue. Since allelopathy is something that in very low cell densities of an allelopath is going to be inefficient or effectively not effective, have no significant effect, how do the populations initially increase anyways? To get to the point where they can have those effects on a competitor through collective toxicity. And there's, I should have the citation here, there's a nice paper in 2009 in PNAS, it's Johnson and all, and they make this point very eloquently using very basic modeling, and basically their conclusion is a toxic alga would need to have, secreting toxins, would need to have a bloom density initially before any sort of benefit of toxicity would actually start to. So, is that clear? So again, making a lot of simplified assumptions, which we'll explore, like evenly distributed toxins and so forth. Okay, so we need solutions to both of these. And this potentially, it's a grainy video, I still haven't been able to improve on it. This might give us a clue. So this is actually a mixed population of the low toxicity small cell there, and then relatively larger and high toxicity cells. And then it's kind of washed out, but this green cell you can kind of see there in the middle there. Just keep an eye on that. Oh, okay. I hope it plays. Again, kind of jumpy video, but hopefully you can see what's going on. So there's one parvum cell really hassling it. But then most of the cells around there are lying kind of quiescent. They're not up to much. Until something happens and they're suddenly really into it. So that's not what you would expect from the unicellular plants, the way that we typically think about algae. I think that the tendency to think of algae as unicellular plants because they do have chloroplasts. I think that might be part of the reason why it took so long. And I would argue still we're having a hard time appreciating ecologically how important this is compared to if they were just poisonous, single-celled plants. Certainly provides an alternative explanation for what the toxins are doing in the first place from an adaptive perspective. This is just showing creosotes down in Tucson, which are allelopaths, keep other plants away so that they can get access to the limiting water out there in the desert. But maybe the other possibility would be that the toxins are functioning more like a venom. That's helping the algae immobilize or kill or consume its prey. Certainly, I think that video, that's certainly what I started thinking about. So in addition to just my own subjective perceptions based on that, watching these things, if you keep an eye on, oh, this is actually, this is just another swarm happening, but so this is a lot like what we just saw. These videos are sped up, I think 4x. But in addition to, so this is kind of like what we just saw, slightly clearer though. Advance. So this is immediately after that little attack we just witnessed. And what you can see, these are the golden cells of parvum, and you can see that a lot of cells are kind of lying around nearby, but they're not necessarily apparently benefiting at all from this. And even though this particular prey was killed by, or it appeared to be multiple parvum involved in this, what I call a swarm, only, I mean, from this video, it only looks like there's really one cell makes off like a bandit. And the rest, you see, I won't goblin it up right there. You see the nice green vacuole right there? And some of the other cells, even the ones that were attached initially, come away with nothing. And it's not a rare occurrence in this species. When you look at them in all different ways, here you can see again the green vacuoles. Oh, and I should say, I've been focusing on swarms. Single parvum cells can solo kill these green algae. That's an important point. They don't necessarily need a swarm. Swarms just kind of happen. And you can see there's green vacuoles here, here, here. And then these are fluorescently labeled, and you can just see the yellow fluorescence there just shows the vacuoles. So in case you still didn't believe me. And so what you see here is this is not a public good. This is not being widely distributed among all the cells. This is something that might be better thought of as a private good, specifically. So the cells that are actually involved in the work may or may not be the ones to benefit, but that's certainly not going to be, the benefit doesn't appear to be distributed evenly to all the cells nearby in the same way that you would expect if we were talking about competition or improved access to nutrients due to reduced competition. So, what happens if we repeat the exact same experiment? Oh, I blew it anyway. If we repeat the exact same experiment, instead of using a diatom, which is armored, that turns out to be very important. So remember, the diatom died, it competed, everything like that. The armor didn't save it, but just by observing, the armor appears to prevent them from being consumed, at least in the conditions we've looked at and the species we've looked at. Somebody's disputed this, but we found no evidence for it. You saw the data before. If you repeat the same experiment that we did with the diatom with this green algae, then you see a major difference. Well, first of all, things look the same for the non-toxic strain. It's still being hurt. You add the green algae in there, they're still inhibited by competition. But things are different for the toxic strain. It grows much better with this potential competitor because it's also a source of prey, a source of phosphorus if you can consume it efficiently. And what happens in the mixture? Well, it might, again, it looks maybe a little bit ambiguous here, but this was the only condition under which we saw cell sizes really, really increase between the start and the stop. We still, again, we're basically waiting to do this, with some sort of molecular markers to really nail it down, but that's certainly consistent with that. Okay, so so this is interesting, and this is not the first, I don't want to make it seem like nobody's ever noticed this before. People have. Back in the 90s, there were the first reports that parvum could eat, bacteria or other algae. So we've known about it for a while, but it's just, this idea of allelopathy seems to be very entrenched. It's very, very hard to overcome the intuition, I think, that comes with this. I'm not actually sure why it's so hard, but people really hold on to it. So if we have basically two very different possibilities here. If toxicity functions primarily in predation, then that's a means of acquiring organic nutrients, whereas allelopathy is a means of acquiring inorganic nutrients, which is pretty straightforward. So we can just test the relative contributions. We can test the degree to which parvum relies on one versus the other as a way of trying to understand better, basically, their fundamental ecological niche, which is something I think that we still don't really have a good handle on. And so in this experiment, for each strain, they're grown under two different conditions. In one condition, they have just inorganic phosphate. And in the other condition, they have no inorganic phosphates, but the roughly equivalent amount of phosphorus in organic form, in form of live prey. So equivalent amounts of phosphorus overall, you know, the question is basically which they prefer. And on the y-axis here is just a measure of preference for organic prey, versus inorganic nutrients. It's a log ratio of the density of the parvum strain grown with prey versus divided by growth with autotrophic nutrients. And sorry, what we see is the highly toxic strains in both years are, you know, appear to very much favor prey. The thing I wasn't sure if we would see is that the non-toxic strains actually grow much better with inorganic nutrients. I guess it's not that shocking, but it's somewhat interesting. So we have a nutrient preference here that's consistent across both years. But is that because of just effective toxicity, or might that actually reflect, or might that actually be a result of differences in actual cell behavior? Since you saw predation isn't something they just stumble into, something they requires some sort of active participation, and I think I'm just being impatient with this. I think it probably was about to work. So this is just showing a time lapse of a parvum, toxic parvum population. You can see initially that they're kind of lying there lazily, and we introduced just a very few of these prey, and you can see how the swarm is formed sort of spontaneously. You can see that they're drawing cells in a little bit. It doesn't appear to be just kind of based on random movement. And if you look up here, this is just showing the contours here just show levels of activity if you introduce a very concentrated little source of filtrate from lysed prey. So basically, anything that came out of the prey when you blow them up is entering from here, and these contours are showing how many cells are acting. So initially they're quiescent, and you can see this wave of activity. So they're sensing something. So they're sensing something associated with the prey. Do we expect to see, so if there's different preferences for one nutrient source versus the other, and chemotaxis is primarily involved in predation, we might expect that there would be differences in behavior as well. So you can pretty much ignore that part. So this just shows the assay that Jess Bauer, pioneered here. It is based on other assays, but adapting it for parvum was a lot of work. You take these capillary tubes and you can introduce, again, equivalent amounts of inorganic phosphate and prey lysate and into two different parts of the same well, and then basically just see how many cells go towards each, the classic chemotaxis assay. Remarkably, in my view, we see that the preference here, the behavioral preference, reflects the ecological preference. It might not seem that striking, but this shows I expected there to be neutrality or something, which is what you would get if it was near zero. But certainly for the first non-toxic strain, you see that there's a pronounced preference for inorganic nutrients. So these very closely related cells that are in the same bloom are oppositely attracted to different nutrient sources, which from a simple ecological perspective is pretty striking to have that level of behavioral difference between these things from the same, that are in the same population. I'm running low on time, so I'll move quickly. But I focused almost entirely on allelopathy, but this is showing a rotifer here. It's showing that this collective toxicity over a smaller scale can also influence other ecological interactions. This is these are parvum cells in 4x speed. Hassling this poor rotifer, I would feel a little bit bad when I see this video. So rotifers, of course, in nature are supposed to be one of the primary microzooplankton that control algae densities. And you can see that the algae are treating it as though it were, a green algae. And so they're with sufficient densities, they pretty quickly kill these grazers, everything up into and including fish, by attacking the fish's gills. So they line the gills and just attack them. The toxin itself degrades very quickly under ambient conditions, which is interesting and nice if you're trying to work with them, so you don't have to worry about anything hurting us. They can't really hurt us, but by attacking the fish's gills in the same way that they're doing here, that's how they kill them. Yep. Another rotifer. It's not film there. Anyways, just to wrap up quickly, so although we found evidence that there is a private good aspect to this this trait. That doesn't mean it's entirely private all the time. As densities increase and as the scale of the effects of the toxins increases, then you might have a situation where there's a increasingly widely distributed benefit to producing these toxins. They could eventually result in a proper tragedy of the commons or a collapse of cooperation, as suitable prey are killed, as they tend to be, in these blooms, and instead the bloom is reliant on the collective toxicity of all the cells in order to keep grazers and things like that down. Yes, they've all established a very nice habitat for themselves, but might there be defectors that, once they accumulate, can actually influence the future of the bloom? Is an interesting possibility that we'll be looking into. Another interesting set of possibilities, these are not monospecific blooms as much as we often think of them as. There are actually other species that can grow alongside parvum, and so we're starting to do work with, this is Phenodesmus, which is widely used in feedstock and biofuels applications. And Phenodesmus appears to be, I don't know about entirely, but quite a bit resistant to parvum, which is an interesting thing to observe, because when you try to culture these guys outside for large-scale biofuels production or something, they inevitably get invaded by other algae that outcompete them, or grazers like rotifers that can mow them down. So by co-culturing, if parvum is harmless to them, and if these guys, primarily require inorganic nutrients, and parvum is good at recycling organic nutrients, there's some possibilities there from an applied standpoint. Maybe co-culturing could be the way to go. And this is just, we don't have to spend time on this. It's just underscoring what I was just saying. Yeah, some species die, diatom, green algae, some species don't die. And they may also play a role in the termination of the bloom potentially. So, okay, and with that, I'll take a few minutes of questions if there's time.
So my understanding, and this is probably partially my ignorance about this, but my understanding has been that the underlying reason why we see more of these toxins is because you get inorganic phosphate going into these systems and then triggering that. But doesn't that suggest that that kind of condition would favor those sort of cheater organisms that prefer? And then cheater may not be quite the right word there, but the low toxin ones.
Yep.
So, given all this, why don't they take over in the
That's a good question. I will say that in general, this species, and it's a generalization people make, certainly toxic eukaryotic algae don't tend to be in the front of the pack when it comes to just growing with inorganic nutrients in general. So this is relative to one another.
Yeah.
But between species differences typically favor the non-toxic species, whether it's diatom, little weedy diatom, or little chlorophytes, things like that. So they'll typically beat things like parvum to the punch, but at least apparently in this species, at least in the lab, but also field studies have shown that immediately before a red tide that was another predatory flagellate, immediately before that, you have a little pulse, a little mini bloom of one of their preferred prey species. So that is a cryptophyte. That's a fantastic autotroph. And so it might not be that it's going directly to them, but it might be, yeah, that basically their ability to then exploit that as it comes up.
Yeah.
That's a good question, yeah.
Could you separate out a portion of different toxins that are invested in public goods versus private goods? Using the cells and quantifying the amount of toxins, or if there's a behavioral switch when they're aggregating versus not, and what they're contributing?
Yeah. Well, that's an interesting idea. Yeah, please.
Well, don't they just like their every screen is completely toxic?
Oh,
They're
yeah.
intersynergies.
Okay. Yeah, that's a.
We think that they're producing the toxins inside the cells, and only the toxic screen are actually.
Yeah. Yeah. So okay. Yeah. So it's a working model. Basically, there's still a lot of dispute about the nature of the toxins themselves. There are people that yeah, I mean, fundamentally, some people say this compound is involved, and other people say not at all. So the nature of the toxin itself is still mysterious from a chemical perspective. Real bummer, of course. Certainly, it appears that whatever it is degrades pretty quickly. And we find when we do our hemolysis assays, so just to quantify the total amount of damage that they're doing, when we do those, we find that basically if you take a non-toxic and toxic cell, the same density, them up, filter them out, and then run the assay on that, they're equally toxic as live cells. And you do the same thing, then it doesn't do anywhere near the damage. So, this is just we're working on it. Our working model is that it might be that there's one of the ideas is that it's a bunch of toxic fatty acids. And so if there's some sort of a secretion of a precursor, a toxic precursor, and then secretion, this is our working thinking, and then secretion of something extracellular that's going to, hydrogen and actually create the toxic products themselves extracellularly, then that would explain you get high toxicity for both. Whereas if you leave it to them, there could be basically behavioral differences more than actual per cell quotas of any one thing. Does that make a little sense? Or
I think so, yeah.
well, it's an explanation for why I can't. Yeah.
Create the covering all the costs of producing the toxin.
Well, yeah, it would come down. And so it's unfortunate to think of it this way, but if like ammunition, I can't go. Someone has a better one. But think of it as ammunition, and there's some sort of a feedback mechanism internally where there's some as it's being used up, then the cost would be incurred. And so if the behavioral side of things is what's determining your ammunition, then that would explain why you still see it cost being at any one moment in time. Does that make? Yeah, that's like I said, it's a working model, but I'm actually very interested to hear if anyone has ideas, genuinely, because I would love to hear any thoughts. How to go about even getting at this. But the behavioral aspect of things, we don't have time to get into. Very, very interesting. It's right in the front of our list of things to get into. Really interesting. Okay. All right. Thank you.